בוקר טוב לכולם, תודה רבה שהגעתם להרצאה. נדבר בעברית, כי פרופסור בוקסבאום מוביל קצת עברית.
אוקיי.
כבוד גדול לארח כאן את פרופסור בוקסבאום. ג'ו הוא ראש המרכז לאוטיזם על שם סיבר במאונט סיני בניו יורק. שנים כבר בנושא חוקר אוטיזם, בעיקר את הגנטיקה של אוטיזם. אחד הפרויקטים הכי מרשימים של המרכז הזה שעומד בראשו, זה נקרא Autism Sequencing consortium. זה בעצם איזשהו פרויקט שנתמך על ידי ה-NIH, שבו מרצפים היום כבר עשרות אלפי משפחות עם ילדים עם אוטיזם, בניסיון למצוא את הגנים שגורמים לאוטיזם, שתורמים להתפתחות האוטיזם. פרויקט שבעצם בשיתוף פעולה עם המון מעבדות מהעולם, בעצם שיתוף פעולה כלל עולמי. אנחנו גאים להיות חלק, הצטרפנו בשנה האחרונה גם לקונסורציום הזה וגם מרצפים חלק מהמשפחות שמגיעים לכאן בפרויקט הזה. והיום תשמעו קצת מהממצאים, אני מניח גם של הפרויקט של האוטיזם סיקונסינג קונסורטיום וגם מפרויקטים אחרים על הגנטיקה של אוטיזם ואיך זה מוביל לפיתוח טיפולים או תרופות חדשות לאוטיזם.
תודה רבה, בוקר טוב. It's very nice to be here. I will speak in English, but I do understand Hebrew. So I'm going to take you through a fairly broad view of autism architecture, what we think the causes are, at least what we know about causes, and I'll give a specific example of a rare disorder we work on. I usually speak quickly, especially when I feel like I don't have, only have 45 minutes to get through 2 hours of information. But I really hope that you can feel comfortable interrupting me if something's not clear, and I don't mind dwelling on important points. Some things are more important to convey than others. All right, can everybody hear me okay? Show me one thing. Okay, here we go. So this picture has become quite famous. It's been repeated by a lot of people, and it kind of tells us what we think we know about the architecture of autism. So what you can do is you can take, this is a national sample from Sweden. You can take individuals with autism, you can genotype them, you can look at their family relatedness, and you can estimate how much of different factors contribute to risk. And this down here is what we call common genetic variation, or SNPs. And that accounts for about 50% of the total risk of autism. And some people say it's not 50, it's 38%, or it's 66%. I mean, in biology, I'm very happy if I get the sign right. If I say plus, so within a factor of 10%, I think that's pretty good. Then there's an area of genetics which is what we call non-additive, so de novo mutations, recessive mutations, and so on. That's about 10%. And this is where all of our discoveries are, as I'll show you soon. And then there's another about 40%, which in the past was called environment. That was just a language choice. It was called ACE, additive genetic environment, additive genetics, shared and unique environment. But environment in those days didn't mean what the plastic in your water bottle, it meant things that weren't genetic. So now what we're calling that is unaccounted for. Some of it is the environment, but it's also certain that not all of it is the environment outside. It could be many other things in there. Mitochondrial mutations fall here and not here, for example, right? So this is unaccounted, right? So more than half of the risk is genetic, but that's not the whole picture. So when you think about this versus this, it's useful to think about these two axes. Every genetic variant that we have in the human population can be either extraordinarily rare, maybe only once in the entire human population, or it can be common, right? 30% of the population has this variant. Okay? So this is the axis of frequency. How frequent is that genetic variant we're talking about? It's kind of low for people, I guess, but what can we do? And the other dimension we'll talk about is what's the effect size? Is it a high-risk variant? For example, there are things what we call Mendelian, which means you have a mutation, you get the disease, there's 100% penetrance, right? And then there are things that are very low risk, increased risk by a tiny fraction of a percent. Right? So when you look at this space, it's pretty clear that down here, things that are both very rare and have tiny effects we'll never find, right? It's just impossible. You know, something's 1 in a billion and the effect size is tiny, you're never going to find them. If you look up here, okay, we're also pretty sure we're not going to find anything in autism up here. This would be something that's common with major effect. If that existed, it would just go away, right? So in fact, in most developmental conditions, we don't find anything up here, right? The exception we all know about is ApoE in Alzheimer's disease. ApoE has an effect size of about 3 to 10, so up here somewhere, and it has an allele frequency of about 20 some odd percent. ApoE falls right here. But Alzheimer's disease happens long after reproduction, right? So there's no control on ApoE the same way it would be if it was a developmental disorder which has reduced reproductive fitness. So we don't expect to ever find anything up here. In fact, if it was there, we'd already find it. If it was both common and major effect, you know, that's easy, right? With modern genetics, we would have found ApoE in about a microsecond if we used modern methods. So where we can reasonably find things in autism and other developmental disorders is in this kind of middle path here. Things that are common with tiny effects. So their effect is so small that they're not really subject to purifying selection, right? Or things that are very rare and can have major effects. And you can also have intermediate things. And these are different methods we use to find these things over here. This is sequencing, and things over here are genotyping. Does that make sense? All right, so talking about common variation down here, things that have tiny effects but are common, that's most of the risk. But we have very few findings. This is a paper that just came out on Biomarkers. This is the first study that has clear, unambiguous, genome-wide significant findings with common variation in autism. It has a total of about 30,000 samples, more actually, 50,000 samples, and it has probably 4 or 5 loci. Okay? So everything we've reported, including me, until now is not replicated in the common variant space. And these are really the first findings. So we're just starting now to make some headway down here. And when you find these things, remember they have tiny effects by themselves. So it's hard sometimes to figure out what to do with that information. It's not impossible. This is a study that came out last year. We took the Sinai brain bank and 3 other brain banks in schizophrenia versus controls. We have a much bigger sample set there. And we could look at all the gene expression in the brain, and we can relate the gene expression to common findings in schizophrenia, and we could show, for example, that 20% of the findings in schizophrenia, and there's over 100, you could show the direction of effect on a specific series of genes. Okay, so we can begin slowly to interpret what these GWAS findings are, what these common variant findings are, but the effect size is tiny, and it's hard to model or understand in a direct way. But that's the future. One thing you can do with all this common variation is you can begin to talk about what correlates with what. And for example, this is in autism, okay? So this study here, it made some significant findings, but it also found many SNPs that were kind of nominally significant. You put all those together, you can talk about the total risk, and you can compare that total risk to other things. So for example, there is a positive correlation. Well, this is actually the surprising thing. There is a positive correlation between autism SNPs and IQ, educational attainment, college, and so on and so forth. What that means is when you look outside the autism population, the SNPs that increase risk for autism are actually increasing IQ, educational attainment, right? And that's probably why we have autism loci in our genomes, right? There is probably a general positive selection for this large number of SNPs that have to do with, brain development, but you can kind of overshoot your mark, right? So this was an interesting finding. It's been replicated now by other groups. So we can begin to talk about the relationship between a specific polygenic risk score and a specific trait or another disorder. ADHD and autism, polygenic risk is highly correlated. So risk for one is risk for the other to some degree. Chronotype, how you sleep, no. Subjective well-being, negative. So the more you have this kind of polygenic risk for autism, maybe the less good you feel. All right. So that's all I'm going to say about this major thing of 50% of risk that we know very little about. And now I'll go to where we've had a lot more findings. Things that are rare and have major effect. Any questions so far? This is where it gets complicated. All right. So, and particularly I'll talk about de novo mutations, which we estimate account for about 3% of total risk in the population. In a family, it's very different, and I'll talk about that later. So you heard about the ASC, and the ASC kind of came into being in 2010, and the context was that they were small studies of about 200 families or 400 families or 800 families that were funded and were ongoing, and we all knew when those started that it wouldn't be enough. It's unlikely to find all the genes for autism with 200 families. And when you put all that data together, okay, which was the total of 1,000 families, 9 genes came out, so you got a lot more putting the data together, right? So there was empirical support. And you could also begin to estimate from that data how many genes there were likely you could possibly find under that model. So this is under the model of 1,000 genes for autism. And this was like when you had no samples, when you had 50 samples, as you increased your sample size, we were pretty much tracking an estimate of 1,000 genes. And it's remained true to today. And again, some people say, maybe it's 800, maybe it's 1,200. I'm like, yeah, it's about 1,000 plus or minus something. So with 1,000 genes, you can be pretty sure that your sample size of 200 is not enough, right? And so ASC was built on the model that we all have data or samples, let's put them together. And let's not wait. Historically, I would publish, and then you'd publish, and then we'd have a meeting, and then we would not agree, and then somebody else would publish, and we'd have another meeting. And 5 years later, we put the data together. ASC was built on the model that we can skip all of that. We can start at the beginning, put the data together now. It's a risk, right? You've collected 200 families very carefully. You want to own them. But there's an obligation we have to the families not to wait 5 years. All right. So the first study came out in 2014 of the ASC. It had 15,000 individuals, 2,300 families, and then a case control series that were very carefully matched. Every sample was sequenced by exome, and we used kind of novel analytical methods which were developed for the ASC to find genes. And basically, the genes fell into those genes in the synapse. There are quite a few of those. And those genes involved in chromatin remodeling, and a few that are involved in transcription. So very big strokes, there were 2 classes of genes that came out. These are the genes that we found in the synapse that were significant or nearly significant. And they're synaptic, but they're not necessarily one kind of synapse. They're presynaptic or postsynaptic, they're GABAergic, they're glutamatergic, right? They have to do with how the brain functions, but it's not a single pathway. You can't just say too much GABA causes autism. You can't say that because most of these genes are glutamatergic. It doesn't mean that most of autism is glutamatergic. We happen to have many more glutamatergic neurons and genes than we do any other neurotransmitter type. And so it just tells you something very broad but not specific yet. The other class is primarily around histone remodeling. So these are, your DNA and your nucleus is wound around histones. And those histones are, both the histones and the histone amino acid tails are modified by enzymes, and all those enzymes, there aren't too many in the genome, but so far about 1/3 of them have mutations that are associated with severe developmental delay in autism. So it's a major source of risk. And basically it's telling you that the regulation of DNA, opening it up and closing it for expression, when you dysregulate that, you get brain development abnormalities which can present with autism or intellectual disability. All right. So that was the ASC paper, and the Simon Simplex collection was going on in parallel. They had their own paper. And then in 2015, we put all the data together. It's 20,000 samples now. And to make a long story very short again, there's a strong synaptic cluster, and there's a strong histone modification cluster. All right? So that's where we were. And now, when you look at this kind of gene list, there's a non-trivial amount of high-risk genes that are clearly implicated in autism, no question about it. So SHANK3, SHANK2 maybe, CHD2, FOXP1, ARID1B, CHD8. These are genes now that you can begin to do something with experimentally, right? So these are genes that when they're mutated cause autism with probably a 50-fold increase in risk. So major effect. So if the population rate is 1%, multiply that by 50, that's a pretty big deal. So it suddenly makes sense now to look at these genes in model systems in a very specific way. Right? And that idea is if you have any disorder and you have a gene or any other cause, you can then maybe model it in a mouse or a stem cell and understand something about brain pathology, which is hard to do in patients, obviously, and then maybe come up with novel therapeutics. Right? So this kind of circular path that was tried in Fragile X has become quite attractive in these disorders where you have a gene of major effect. So the gene I'm going to talk about is that one there, SHANK3, okay? And the disorder is called Phelan-McDermid syndrome. I can explain why. It's shortened PMS, and the Family Foundation, PMS is also a menstrual syndrome. And so their tagline is, it's not what you think, right? So it's a genetic disorder, and it presents with intellectual disability and autism. And it all traces to a single gene, SHANK3, and I'll talk about that extent. So there are point mutations in SHANK3, and there are deletions in SHANK3, and both cause the same phenotype. This is, we looked at 17 patients with point mutations, so the biggest cohort prospectively. We also did a literature review. We found 60 mutations described in ClinVar. About 50 of those had some clinical data. So this is the map of all the known mutations in this gene, all which present with autism or ID, and often both. And this is the really surprising thing for many people, that actually SHANK3, or PMS, which you may never have heard of until this minute, is more common than Rett syndrome, significantly more common than Rett syndrome. It's probably approaching Fragile X in its frequency, okay? But it's just unheard, right? Fragile X we knew about decades ago, so we've all learned about it. Rett syndrome we also knew about quite some time ago. We have all heard about it. There are emerging genetic syndromes, DDX3X, FOXP1, ADMP, this one over here, which most people have never heard of, and they're at least as common as Rett syndrome, if not more. Right? So the genes are coming out, and they're not really making a transition to the general knowledge view because there's kind of too many of them. All right, so I'm going to say that about 1% of autism and 1% of ID have mutations in this gene. If you look at people with both autism and ID, it's probably higher. So, it's a fairly significant gene. All right, so back in 2010, we started to see families with mutations in this gene, and we did the first prospective analysis. Families come in for 3 days, they get a full phenotypic workup, and we can start talking thoughtfully about what we see in the kind of neurological and psychiatric domain. And everything till then was kind of case reports and literature review. So you can see that, 85% met criteria for autism. You can see that the IQs were quite severe, right? Severe to profound intellectual disability for the most part. They had significant language issues. Their receptive language was slightly better than their expressive language, both fine and gross motor skills. Nothing specific. These are things we see, throughout the spectrum. Okay. One of the things that's interesting about this, and I think it's a very important point, is that these kids they're not your kind of classic Canner autism, and I would say that about half of autism is not your classic Canner autism. They're not really socially averse or completely immune to social cues. This is when you put somebody in the scanner and you play communicative or social sounds versus non-communicative or non-social sounds. From the time you're born and even before, a certain part of your brain will activate preferentially to social sounds compared to non-social sounds. And these kids in the scanner look perfectly typical. Okay? So they're social, they're kind of, all the networks, at least with auditory social sounds, are preserved. In idiopathic autism, it tends to look like this. It's not perfect because idiopathic autism is not really a single thing either. But if you average a lot of idiopathic autism or Canner autism, certainly, it looks like that. In other words, some of the basic wiring for social cues is missing. But these individuals, and my guess is about half of autism looks more like this than like this. These individuals have everything in place. They're just profoundly disabled in terms of their attention, in terms of distractions, things like that. And so they're not going to make a good reciprocal social, they're going to have deficits in reciprocal social interaction that meet criteria, but it's not the same thing. And I think that's not a non-trivial point, right? Invariably, people lump all autism together, and it doesn't matter if it's animals or patients, right? In animal models, you can't write a paper on an animal model with autism without saying there's social deficits, which is ridiculous because mice are not humans, and even in the humans, a certain proportion don't meet criteria for autism, and why should every mouse show a criteria? And here, some of the social network is preserved, right? So it hurts the basic science, and it hurts clinically because clearly there's something very different neurobiologically between somebody who does not respond to social cues or finds social cues aversive than somebody who responds to social cues just can't meet criteria. Right? I think the classic example is if you take Williams syndrome, which is a hypersociality syndrome, half of those kids will meet criteria for autism because they just don't do the reciprocal, they will talk to you, they'll get in your face, I don't know if I'm sure you've seen, so they're just like, they're this close to your face all the time. They're hypersocial, there's no question about it, but they don't do a great job at turn taking, right? So they meet criteria for autism, but it's got to be a different mechanism. And just lumping it all together is not helping anybody. Anyway, so here's one of the strengths, right? So there's that. This is something we talked about yesterday. This is, as you know, our ability to kind of tabulate or capture sensory issues in autism is fairly crude, and we tend to lump together, again, hyper versus hypo, right? We just say it's a processing disorder. Again, neurobiologically, it's unlikely that what makes you hyposensitive is exactly the same as what makes you hypersensitive. We should probably start to separate those. It's also possible that you might be sensitive in one domain versus another, okay? And most of the people coming through the center who are coming through, for example, Phelan-McDermott syndrome, are nonverbal. So we're kind of stuck. It's parental report, which is okay. The parents can say, yeah, very sensitive to, shirts or things like that. But wouldn't it be better to have a measure that, like the ADOS, is observational? So we spent some time developing a toolkit, okay, where we challenge the kids with visual, tactile, and auditory stimuli, and we've developed a system of rating, and we've shown that it's reliable. And now we can say, and we compared it to existing scores. We can say somebody's hyperreactive, generally, hyporeactive, or seeking, so the 3 DSM subgroupings of sensory processing issues. We can split it into visual, tactile, and auditory, and what you can see is this is typical versus autism, pretty severe differences, but some are hyporeactive, and some are hyperreactive, and some are seeking, just like DSM says. And again, that's probably very different mechanisms. Probably better to not put them all together. And here's Phelan-McDermott syndrome. So just look over here. Typical developing, looking at hyper, hypo, and seeking. ASD, more seeking than the other two. PMS, primarily hyporeactive. These are the kids who, they come into your office, they smack their knee really, really hard on the side of the table, and they show no sign of anything. Or their parents will say they burnt their hand and they didn't respond, This is a pretty profound hyporeactivity. And it also happens in autism, right? Not in everybody, but there's a fraction of autism hyperactivity. We're doing biomarkers. EEG is one of them. This is a visual evoked potential. And just look over here. This is typical developing. So there's a checkerboard screen. It switches. And after a certain amount of time, you get this kind of deflection and then an up in typical developing. In autism, we'd already known it was kind of blunted. Okay? And in PMS, it's really, really, that first peak is almost gone. Okay? So we can start doing kind of EEG biomarkers. If you knock out the gene in mice, you actually see the same thing. So this is the patient, control Phelan-McDermott syndrome. This is the mouse, control PMS mutation. All right, so it's a translational biomarker, and what you really want to know is it predictive, right? if I change this in the mouse, does it mean something useful to changing this in the animal, in the patients? You can see where my head is. And everybody says, okay, that's 1% of autism, great, but what about the other 99%? And I'm very sensitive to that. I don't know if we can actually start with a 99% and find treatments. I'm actually skeptical, in terms of pharmacology at least. So I'll be okay if I retired and I said we help 1% of the population with autism. I used to work in Alzheimer's disease, and I moved out of it, and I was sitting on a bench in Cape Cod in the Marine Biological Laboratory, and one of the biggest names in the field came up to me and sat down. We chatted for a little bit. He had retired 2 years ago, and I realized that he had spent his entire career, and the needle really hadn't moved in 30 years of hard work. Now, I hope he doesn't see it that way himself, because on his shoulders and the next generation that we will make progress in Alzheimer's disease, but his entire life dedicated to Alzheimer's disease, and really dedicated. I knew when he was an older individual and he was still working 80 and 100-hour work weeks. He would get up early in the morning and drive to a parking lot and have meetings on the weekends with his lab just to keep the things going. And I felt like, I don't want to be there, right? So if I can make a difference for 1%, I'll feel pretty good. But it's also possible that what we learn in PMS may apply to a part of the idiopathic autism world. So this is that same EEG that I showed you earlier, focusing now on this first bit over here. So I said this is typical, this is autism and this PMS, but this is actually quite a broad peak, okay? And some look like this and some look like that. As is always the case in idiopathic autism, the variance is high, and somewhere in there are people who look more to one side or the other. If you look at your PMS values and you kind of take a pretty harsh cutoff, okay, you'll see that about 30% of idiopathic autism look like, if you will, PMS. Doesn't mean that it's the same mechanism, but at least it's more likely that the neurobiology here is more similar to here than the neurobiology here. Okay? So we can use biomarkers from rare disorders and at least potentially use them to subgroup larger groups of autism. And so the argument then would be, if a drug works here, maybe it'll work in this subgroup here. So I don't think we have to give up, even if we're focusing on rare disorders, we have to give up on idiopathic autism. It's just an entree point into the broader spectrum. All right, so just like some more facts about what we know about the genetics of autism. First is autism and intellectual disability and even severe developmental delay, so before you even have an IQ, you have severe motor disability and so on and so forth, there's a very significant overlap of risk. Not surprising, perhaps. We can, it's not trivial, but we can actually find genes that are more autism-like and more ID-like, even though most of them are probably both. And what's interesting is the odds ratios in intellectual disability are much higher. This is a more severe disorder. These are kids, as I said, it's not even ID, it's, some of them they got to a clinical geneticist before they even had an IQ measure because they're showing profound global developmental delays. So not surprisingly, the odds ratios of loss of function in this group of people is much greater than in autism, both for loss of function and missense mutations. If you put all that together, the autism findings and the developmental delay findings, it definitely maps to corticogenesis, the development of the cortex. Again, not in the most beautiful single pathway way, but many genes that we know are involved in specifying the layers of the cortex and making specific kind of projection neuron subtypes are mutated in autism and or ID. I think, thank you. I appreciate it, and I should probably have some. So this is kind of just being human. It's about developing kind of, the social architecture and the cognitive architecture that makes a human being a different species than another species, right? So autism is part of who we are. This is what I meant by the odds ratios. The top genes have an odds ratio of maximally about 80, which is effectively Mendelian, between 20 and 80. But very quickly, we come down to odds ratios of 5 and lower. So the number of genes that are Mendelian in autism is, a couple of dozen. Okay? So all the big genes, at least on the autosome, we've already found, because there aren't too many of them. X chromosome, give us some time. Recessive, give us a little bit of time. Even there, we found probably many of the top genes. So we're not going to find a series of rare genetic disorders. That's clear. We're going to find a few rare genetic disorders and then genes that increase risk by some significant amount. So, we're going to find, well, I'll stop there. And then here's the question that, this is 50% of risk, and here's another, 10-ish%, and here's another 40%. We have a lot of findings here. What do we know about how this interacts with this? Okay, and we can actually make estimates from that. So if we have a sample that has genotype data plus all the information you have about de novo variation, you can actually ask about the interaction of these two things within a family. And here's the important thing. The polygenic risk is everywhere. Within a family, if there wasn't a de novo mutation, 80% would not have autism if it was a CNV, and 60% would not have autism if it was a loss of function mutation. In other words, the family has this throughout the family, and then the child has this in addition. So it's an additive genetic effect. And you can also have autism without this. It could be just more of this, or it could be this plus this, right? So that's the additive model. Things just build up. And it could also be this plus something in the environment, right? But this substrate is probably almost universal. It's not absolutely necessary. If you have a 100% penetrant gene up here, you don't need to have family risk. But most people don't have that. They have this plus something else, all right? And that interaction is what makes a family. The reason you can give genetic counseling to a family, even though they have this, is because when you find something, you can say, if you don't have that, your risk of autism has gone down by 80%, right? So you can give genetic counseling even in the context of polygenic risk. The second thing or third thing or fourth thing we know about autism, these are some of the genes I showed you that we found in autism. Here's a paper that came out a year before that one in congenital heart disease. So here are your histones again. Here's the DNA. We drew it much more crudely. They drew it much more beautifully. But the genes are completely overlapping. Okay, so genes for CHD overlap with genes for autism. In a certain way, it's not surprising, because if you look at congenital heart disease, there are often neurodevelopmental outcomes, right? If you look at brain genes, are also, VCFS is the classic example, DiGeorge syndrome, right? With DiGeorge, we've known for decades increased risk for, autism, intellectual disability, schizophrenia, epilepsy, and structural abnormalities, congenital heart disease, So it shouldn't be that surprising, because that is our classic genetic finding in the past. But it's really important to know that there's no one-to-one correspondence between DSM-5 and any gene. It's a many-to-many relationship. For every disorder, there are many genes, and for every gene, there are many associated clinical findings. Okay, so it's just important to pay attention. The good news is, if you find a treatment for CHD8, maybe the people with that mutation, whether they have autism or multiple congenital anomalies, you can help all of them. Maybe that's a way to look at it. All right, so I want to talk just on 2 slides about this over here. So I said it's not just environment, but a lot of it's environment. And here's where, taking large population registries or medical records from a hospital can help to dissect out risk factors that are non-genetic. This is the Minerva study, which includes a cohort in Israel. 27 million people in a registry-based analysis of autism. And you can begin to talk about, different environmental effects of the vaccine, increases risk by 0.000. Some of the things you've heard about, birth trauma, SSRI use, increased odds ratios. One of the important things about all of this that the press always loses sight of is that, if SSRIs are associated with a twofold increased risk in autism in the child, SSRI use by mom, it doesn't mean the SSRI caused autism. It means that there's an association. And what's the thing we learned first in science? Anyway,
It's not causality.
yes, causation does not imply causality. Association does not imply causality. So, you know, mom took an SSRI for a reason. I hope. I hope it wasn't just for fun. I've already shown you that ADHD risk and autism risk overlap. In fact, every psychiatric disorder in terms of polygenic risk space overlaps. Whatever reason mom took SSRIs, all by itself increases risk for autism in the offspring. Sorry to say it. It's not the SSRIs, and if you control for moms for indication, this goes away. It's hard to do in California, where these studies often start, because the California registry is not that good. It's nothing to do with California. It has to do with the state of the healthcare system in the United States of America. If you go to countries where there's a robust kind of medical records, Israel is a good example, you can actually control for indication. You control for many other things. You can even control for compliance. If mom didn't go back to the drugstore and get her refills every X number of times, she wasn't taking the medicine. It's not, you know, you can't do that in the U.S., right? Birth trauma, same thing, right? I have no doubt that hypoxia is going to contribute to severe and developmental outcomes. However, I can also tell you that if you look at the siblings of individuals who had birth trauma and autism, their rate of birth trauma is higher, perinatal events is higher, even if they don't have autism. There's something going on in the family that increases risk for both perinatal events and autism. Right, we talked about C-sections. One reason people have C-sections is because there's hypotonia in the infant. Hypotonia in the infant is associated strongly with autism. So, it's hard, you know, making causal inferences is important, hard to do. There are ways to do it even with epidemiological data. There are elegant ways to do it with genetic data with epidemiological data. All right, this is something we just came out with. It's about maternal effects. So maternal effects are things that happen in the maternal environment, the inter-unit environment, external mom's choices in life, smoking, non-smoking, dieting, not dieting, that affect the child, but not through a direct genetic effect. And the reason we looked at this is because there's been a lot of talk about maternal effects in autism. They've been called different other things. Maternal immune activation is a maternal effect. So you've heard of MIA. There are lots of them. And the data for support of them is not the strongest, but there's a direct way of measuring whether maternal effects are a major part of the spectrum. And basically, you take advantage of the fact that there are different kinds of half-siblings and different kinds of cousins. If maternal half-siblings are more alike than paternal half-siblings, or vice versa, that's an evidence of a maternal or a paternal effect. Okay? With multiple sclerosis, half-sibs related by the mom are much more likely to share the trait than half-sibs related by the dad. That's a classic maternal effect model, and it's also true for cousins. So if you have a big data set, you can ask about whether this is different from this or this is different from this, and the short answer is it's not. Under multiple models, there's zero evidence of maternal effects in autism. It doesn't mean there are no maternal effects. It means it's a tiny part of risk architecture. And we also can't look at sporadic events that happen once to the mom during pregnancy and never again. But if mom had an immune disorder that was it by itself increasing risk for autism, that immune disorder will also be in the second pregnancy, and we'd see much more correlation than we do. So we're pretty sure that some of the common things that we think about in terms of autism, like blaming the mom, not true. All right, back to the ASC, and I'll wrap up pretty quickly. I'll talk about where we're going today. Oh, I haven't even gotten to part two. Jesus. No. So this is the ASC today. We now are up to 30,000 today, and we're going to have 50,000 pretty soon. This is the first publication. This is the publication that came out in 2017. This is the current data freeze. So this data we got cleaned up end of last year, middle of last year, and sorry, we got it finished and cleaned up now. And then here we are in terms of sequencing, and BGU just got sequenced August 17th, and I'll send you an email how to get the data. I thought I did already, but so this is not in the current data freeze, but it's pretty close. All right, so I lost a bunch of slides. Okay, well, I'm going to say in words. I don't know how that happened. I'll say in words what I didn't show you about PMS. So with Phelan-McDermott syndrome, I've shown you what you can learn about kind of from the mouse. What I didn't show you is in the mice, we found that IGF-1 was helpful for the mice. IGF-1 is a growth factor. We chose it for a weak biological reason and a strong pragmatic reason. The biological reason is IGF-1, given even peripherally, is known to enhance glutamate synapses. And SHANK3 deficits, we showed very clearly, in the mice have weaker, more immature synapses. That's the biological rationale. The more pragmatic rationale is that IGF-1 is very, very safe, already used in kids, and we knew that if we had any finding with it, we could do a clinical trial. And so from the mouse, we did a clinical trial of IGF-1 in PMS, and we saw some first positive findings in a tiny study. We just finished another study, and some of the findings hold up, not all of them. And we're hoping, we scratch our heads, but we're hoping that the company will give drug for free for a multi-site trial. The drug is very expensive. It costs $14,000, even for a 3-month trial. So that's kind of where we're going from the mouse biology to a drug to trying it in patients. The second example is that together with folks at the University of Haifa, we've shown that there's also a clear deficit in oxytocin signaling in these animals. So oxytocin, we think, generally involved in social behaviors. The data in autism is kind of mixed, but if you look at these animals with the SHANK3 mutation, the oxytocin system is really broken. Not like idiopathic autism, like specifically broken. And so putting oxytocin in those animals corrected not only their behavioral deficits, but also their attentional deficits, which is it was the first time that attentional deficits were shown to be improved by oxytocin. So again, it's easy to do an oxytocin clinical trial in this patient population because it's a very safe drug and so on and so forth. And so now we're doing a trial of oxytocin. These are not the top drugs. It's what we can do as an academic center. But we think that that model that I showed you before, that if you have gene mutations and you study the biology in animals, you'll come up with, understanding of biology, and either through using existing drugs or ideally through novel therapeutics, you can actually make an improvement in the mice, and then you can go to clinical trials. And we think that if we kind of stick to that in autism, in the rare genetic disorders in autism, we'll have some progress in the field within a decade. That's our hope. All right. I kind of lost some of my most fun slides that I do with mice and rats, but I think I got the big picture done. I'll take questions now. Sure, starting at the front.
I have one question. You didn't speak directly about specific findings of DNA modulation, methylation, or Histone methylation.
Yeah.
Is there a way to go through this to find targets that are repeating in autism?
Sure.
Or?
Yeah. So, well, I can't bring up a slide because it's not my computer. There's a really nice series of genes that are clearly involved in neurodevelopmental disorders that are histone modifying or reading enzymes, and all of them affect the expression of other genes. So it will be very nice someday soon to knock down those genes in vitro in human neurons in culture, for example, and begin to ask, are there overlapping target genes, and see if we can't, come down to primary pathways or a few primary pathways. I think that's an experiment that needs to be done, and who's going to do it? I don't know. We spent a lot of time with shRNA trying to get it to work on a large scale, so we can knock down 100 genes, serially, quickly. Now the new thing is not shRNA anymore, so people are trying it with some CRISPR silencing complexes, but I think that experiment will be very informative. So I think that's going to answer a question that's probably a little easier than these diverse synaptic genes. If we just take all the 15 or 20 histone modifying genes, do they appear to kind of dysregulate similar pathways at any time during kind of, nerve cell development? Yeah. That answer the question, sort of?
In the mice model, yes.
Yeah.
In human, I guess.
Yeah, well, human,
it's a different way to go.
Yeah, so humans is going to be tough, right? I mean, we don't we don't have good human samples at all levels of dysregulation. I think the mice is the right way to go, or human neurons in culture, right? iPSCs. And then once we find things, we can ask questions, targeted questions in humans. Seems to me. Hi.
I have a question regarding that common variance chunk, which is fascinating to me. Do you think that inside this chunk you will find different pathways, different groups of effects, or would you say that this is just a simple additive? I mean, I would assume that there are different types, and then you would.
Yeah. So we don't have enough to even begin to answer that. I suspect that not everybody has all of them, and there probably are different pathways. But how to understand it is kind of a mystery, right? So I say that polygenic risk for autism overlaps with ADHD. Is it all of the SNPs, or is it a subgroup? There's always the problem with most genetic samples that we don't have really good comorbid information. So is it heterogeneity or is it pleiotropy? We don't know that either, really. We have some ideas, but it's not really solid. One of the things that I think about, which is a different way of looking at it, is if, DiGeorge syndrome is associated with epilepsy, autism, schizophrenia, intellectual disability, are we just doing really bad diagnoses? Or are these really distinct entities, and why does the same risk factor produce distinct entities? So maybe that's what the polygenic risk is doing, right? That within the polygenic risk, you can be more schizophrenia-like or more autism-like, so they're not fully overlapping. Even if I say they correlate highly, there's still differences. And so maybe the polygenic risk could be with sculpting, if you will, what happens when you have the DiGeorge mutation, right? That, it has a neurodevelopmental outcome, but the exact way it lines up is much more defined by polygenic risk than it is by the major hit of 22q11 deletion. So that's how I've been thinking about it. Just a speculation, not yet testable, but it's, I like it because it answers the question of why are they so, why is it congenital heart disease and autism, same gene, right?
Do you think that then the environmental effects could be the ones that are
Sure,
steering?
Of course, yeah, yes. The answer is, of course, it could be, and it's probably part of it. I favor some of the genetic findings are recurrent within families, and they tend to all, be more or less true. But it's not. Yeah, it's a weak argument. I'll shut up. Yeah, could be.
Thank you for really fascinating talk, and I'm sitting here as a clinician and finding it quite confusing because the DSM-5 takes us to lumping autism. And
big mistake.
Big mistake, but that's for the next many years, I guess, by the time they come with a new edition. So we are really lumping all the autistic kids into ASD, and the hyper and hypersensitive goes together. So it's, what should we do? And I'm thinking about wanting to promote research even here in a smaller scale. What do you do with all these?
Well, I mean, we spend a lot of time reminding people that it is a heterogeneous disorder, not only genetically, but also obviously clinically. And I feel like DSM had a pretty severe, unanticipated side effect, right? People, when you go to every meeting now, there is a community of advocates, self-advocates. They're self-identified as Asperger's syndrome individuals. They're very vocal anti-science and anti-research. They scream and shout. That's half of the audience, and the other half of the audience is a family that has a very severely affected family member who want there to be more research and more treatment into whatever their kid, brother, dad, not dad, but whatever their family member has, because they know that individual is severely disabled and suffering, right? And so by lumping Asperger and autistic disorder, we've created this kind of sense that it's all one thing, and that they should be treated, that somebody with Asperger's is a reliable spokesperson for a disorder that because they have Asperger's, they actually can't really be empathetic towards, right? And vice versa. So I think that's a very severe limitation that DSM has done. It's really hurt the field enormously. I personally think we should go back, of course you can just kind of qualify autism with intellectual stability and language delay. That's kind of a pain in the ass, but I think it would be good to go back to autistic disorder. I think it would be good to think about counter autism versus, or more kind of break it down into whether you're socially averse or socially not aware versus other things. You're not going to make progress research-wise, even in treatment, even kind of clinical treatment, if you just say it's all one thing, right? Even if we just do that, maybe oxytocin does work for those where there's real social kind of neutrality or aversion, but doesn't work for those that have other things, and maybe we should just, cut that group up into 3 and see whether oxytocin works better in the subgroup, right? So I think I think the field won't advance until we do a better job of splitting, and it's an educational process for everybody, right? I mean, clinicians have to kind of embrace it, families have to embrace it, and the funding committee has to embrace it. I don't have any great ideas.
About the social pragmatic, the new diagnosis that was thrown into the basket, anything. Can you relate to that in a way? Do you see it with social pragmatic?
Not really. I mean, I think it's whatever it is. It's not a major. It's a tiny slice of the pie, right? I mean, you know better than me, but do you think that's a major? Yeah. Yeah, I think it was a kind of a kludge, what they call a kludge in computer science. Some kind of a short-term attempt to fix something that doesn't really make much. Didn't really have a strong impact. Yeah, it's a real issue, right? I mean, just when you talk to neurobiologists that we've lumped together hyper and hyposensitivity, they kind of go like, what now? Right? There's no way, right? They just look at you like,
Well, I have something in their defense, that sometimes the same children show hypersensitivity in one context and hyposensitivity in another context. No, within the same children, they can be...
Kids that have both.
That they're...
No question. But that's why you shouldn't lump together all your sensory things. I mean, in a certain sense, it's more sensible to lump sensory because there are some common mechanisms than it is hyper versus hypo. But sure, wouldn't it be better to parse them out into the sensory modalities and into the direction of the effect?
And lumping communication and social cues.
Yeah.
It's an overlap in some areas, but...
Right. No, I think it's, one of the things I always say to neuroscience students is, you're a great neuroscientist, you know about the brain, tell me how social deficits and repetitive behaviors, where in the brain do those come together? I mean, clearly they must, but we don't have a mechanism that easily puts them in the same kind of CNS space, right? So it's good to lump sometimes, They say all historians are broken down into lumpers and splitters, right? And I think we have to be both, but I think we've done a little bit too much lumping. I think it's not gonna help the field. But I think, and genetics is just one way of splitting. There are much more thoughtful ways. I'm just a simple-minded guy, right? EEGs, phenotypes. There are ways of splitting. A careful assessment of what kind of autism it is probably the best way to split it. It's not using ADI and ADOS and doing factor analysis because that's been tried ad nauseum and didn't do anything. It's about asking questions about the exact kind of aspect. What does the social deficit look like? Are the repetitive behaviors higher order or lower order, right? These are things that a clinician can very easily kind of parse people in a thoughtful way, and you've already got some reasonable subgroups, right? And it may well be that oxytocin or something we've already tried works in 1/6 of the population that you can clinically do a reasonably good job of splitting. I'm clinically using standard instruments, but also, of course, using kind of things, SRS, things that are, not necessarily part of the formal clinical evaluation, but are clinical tools. Yeah. More questions?
What's funny is that those clinicians decide if they will be in the research or not.
Right. So that's those clinicians. I mean, and that's an issue for families too, right? Clinically, like people who do research, they want to have a tight diagnosis, right? The world isn't that clean, but it's often usually better to do a study in a more defined group. But you always have to go broader, right? The Ivory Trial of Academia, does all sorts of wonderful things for the people that come to their center, but if it doesn't kind of reach out into the broader community, including the gray area. So we have a social skills program that we piloted only on people that met, ADI and ADOS autism and DSM-5 or DSM-4 at the time, and as soon as the moment it went out into the real world, there was no exclusion criteria, right? When you're working in a community center, a JCC in that case, and a family wanted to join in the social skills training group, nobody would or could say, let's do an ADI and ADOS before we can let you in. So that population is much broader. There are kids with ID, there are kids with, questionable autism, there are kids with some kind of combination of cognitive disability and ADHD, all the things you see in a kind of a real population setting. So we actually don't know if any of our interventions, as soon as they go out into the real world, are as effective as they are in the ivory tower. I still think it's worth, at the outset, in order to even see if it works at all, trying to be somewhat prescribed. But I think right away we have to then say, okay, enough of that. Let's go to the broader community and see if it still works. So, the social skills thing was deployed across the New York area, and very quickly I said, okay, that's fine, but let's also do, let's treat it like a study too. Let's do pre-post on everybody and see if things like the exact diagnosis and cognitive levels and so on actually make a difference in the real world, right? So I think we always have to think about the next step from an academic medical center to every other part of the universe, which is everything else. Yeah? Okay, thank you.